Hello, this is Seher from Easy Peasy, and the topic we are going to discuss today is called Telomeres and Telomerases. Telomeres are non-coding DNA that are located at the end of the linear chromosomes. Look at the picture. You can see this is the cell having a nucleus in it, and inside the nucleus we have chromosomes. The end portion of this chromosome that is showing in red color is basically the telomeres. The DNA that is present over there, they have non-coding regions there. It means they don't have any kind of genes that is necessary for our survival. In human being, we have approximately 8,000 to 10,000 nucleotides long telomeres in our chromosome. If I talk about prokaryotes, in prokaryotes, the DNA present in it is mostly in the form of circle, just like in plasmids. So in prokaryotes, we don't have any telomeres. But in eukaryotes, because we have a true nucleus there, and nucleus does have chromosomes in it, and the DNA present in those chromosomes are linear, so we do have telomeres in eukaryotes. Now, if you can look the telomeres at the end of this portion, you can see that telomeres end portion is not equal in size. One strand is longer than the other strand. Now, when I look at those strands closely, this is basically the lagging strand that is longer than the leading strand. And the nucleotides present in the telomeres are repetitive. So they have the sequence that is adenine, adenine, thymine, cytosine, cytosine, cytosine in the leading strand, and in the lagging strand, the complementary nucleotides will be thymine, thymine, adenine, guanine, guanine, and guanine. Now, this particular sequence is going to repeat itself again and again in the whole telomeres region. Now, this sequence is called as non-coding hexameric sequence. Now, look at this part. This part has single-stranded DNA. Now, DNA cannot exist in this form because if it does exist in this form, the enzymes called nucleases can come and attack this single-stranded DNA, and it's going to get degraded. Now, this single-stranded DNA is going to coil upon itself and make a loop kind of structure that is called as T-loop, like this. And that single-stranded portion that was visible there is going to hide itself in this tiny portion, and that is called a D-loop or displacement loop. Now, why the telomeres are making the T-loop there? Because this is a kind of knot for us, and it's going to prevent the telomere ends from being recognized as the break point by the DNA repair system. So DNA repair system cannot recognize that there are some break points in it and cannot make changes in this T-loop structure. Now, this T-loop structure needs to be maintained in this form or in this configuration. For that, we have 6 group of proteins that is present there. And these group of proteins together called sheltrin, and they're going to attach to this T-loop. Like I just showed one group here, but there are several different groups present on the whole T-loop and maintaining its structure there. Now, the question is, why the lagging strand is longer than the leading strand? Just assume that both strands are in equal size. For leading strand, we are attaching a primer followed by this daughter DNA nucleotide strand. So we don't have any problem with it. For lagging strand, because it is present from 5' to 3' end, we need to make small different fragments of DNA called Okazaki fragments, and then these Okazaki fragments join together to form the daughter strand. So let's see if this is the primer. This is the first Okazaki fragment. Now I'm removing the primer and I'm attaching it at the end. and I'm making the second Okazaki fragment here. Now I'm removing the primer again. What about this portion? This portion needs to be replicated, but I don't have any place to put the primer back again to attach nucleotides with it, right? So a particular part of DNA is not replicated and we're losing genes on it. What if the genes present on this parental DNA on this specific portion is necessary for our survival, right? So if those genes are not replicated in the daughter strand, we cannot survive in this world. So for that, the telomere comes in handy, and that's why the telomere lagging strand is longer than the leading strand, because the telomeres have non-coding sequence there. So if we are losing a little part of telomere there, we are okay with it. The chromosomes are okay with it because they're not losing any genes, right? So this shortening of telomeres in every cell division is called as telomere shortening. So for every cell division, we are losing tiny portion of telomere every time. Now, once the telomeres reaches to a point where we cannot divide or lose telomere again, because up till now in this picture, if I remove the telomere part again, I might lose my real genes as well, and I cannot afford it. So at this point, the cell stops dividing, and that process is called as senescence. Now, this cell or this particular chromosome, it either remains within our body, but they're not dividing anymore, or it can go in the process of cell death or apoptosis. Okay? Now, when the cells are killing itself or going in the process of apoptosis, we are going to see some symptoms of aging. So aging is basically related to the shortening of telomeres there. Make sense? Okay. Now, we cannot afford losing telomeres every time in some specific cells, and these cells are germ cells or stem cells. We cannot afford that. For that, we have a helper protein, and this time the enzyme we have to save this telomere is basically called telomerase. Now, this telomerase is basically an enzyme having two subunits called TERD and TERIC. The TERD is basically the protein part of this enzyme, and it's gonna do the function of reverse transcriptase. The word transcriptase means turning DNA into RNA. And if I use the word reverse before that, it means I'm doing it oppositely. So I'm turning RNA into DNA structure. Make sense? The second subunit that is TERIC over there is basically the RNA, and it's gonna work as a template for telomere to attach more nucleotides to complete its length. Now, how it's gonna work, let's see. Now, this is the telomere we have, and it is shortened right now. I'm going to put the sequence that is repeating itself. So in this lagging strand, I have sequence called TTAGGG. Now I'm putting that telomerase enzyme here. This telomerase enzyme is gonna work as a template because RNA strand is present inside that telomerase, and it works as a template here, attaching DNA nucleotides further to complete the length of telomere again. So with adenine, we will attach thymine again, then thymine, then adenine, then guanine, 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 and so on and so forth. Okay, so if we have the enzyme called telomerase, we cannot lose telomeres in cell division. Make sense? Okay. Now, once the replication is done, it's going to coil upon itself again to make the T-loop, right? So once it's gonna go for this process, I did mention it in my last video, when the process of replication is gonna end, they're gonna make the hairpin kind of structure. So when it's gonna making the T loop there, the initial structure looks like the hairpin structure over there, and that shows us that the DNA replication process is terminated now. Okay. Last but not least, we have telomeres directly connected with cancer cells. How? Look in this picture. This is the normal cell having a normal chromosome. This yellow portion is basically the telomeres we have. Now, telomere is getting short after every cell division. So once it reaches to the critical point, these cells are going for the cell death, or they're gonna halt the cell division or stops the cell division there. But if you have cancer cells, there are some kind of mutations present inside the cell, and they're gonna ask the telomerase enzyme to recover the portion that was lost in these cells to make it again. Now, telomeres are making again, it's not becoming short in these cells. That's why the cell cannot stop cell division, and it's going to divide again and again and form tumor inside the body. I hope you like this video. If you did like, hit the button like. Thank you very much for watching this video, and please subscribe my channel. Thank you. Bye-bye.